This is Coast to Coast AM with Art Bell from the Kingdom of Nye.
It is indeed my guest, Dr. Jonathan Reed, Robert Wraith, and Dr. Harold Chacon, a microbiologist. And once again, so that you know, reading from what was sent to me, Dr. Chacon is a degreed microbiologist graduating with honors from Catholic University of Puerto Rico with a DS Master's Program Certificate in the field of microbiology, biomedical research and cell immunology, biochemistry, and zoology, currently studying and residing here in the U.S. And I'm not going to run my commercial break now. We're going to go directly back to our guest. And Doctor, if you would continue, please, the tissue sample you said was not, had similarities to, but was in fact not human.
Not human. Okay, it was about 3 millimeters in thickness, very, very fibrous consistency, and the surface was kind of light grayish brown.
Really?
Extremely transparent, much more transparent than human skin, much more.
Okay.
There was no signs of any hair or hair follicles within the surface of the cell structure.
Right.
Absolutely no signs of it. We found some subcutaneous tissue, that's the one that is under the surface, and that one has some sebaceous-like glands. Those are the glands that produce usually fat, but half as many as we found in the human skin.
Half as many.
Half as many.
All right. Are any of these findings consistent with any other living mammals on Earth that you know? No?
No. Probably the one that could be most related, probably, in some ways, not completely, could be the reptiles.
Reptilian.
But not completely.
But enough difference so it definitely was not earthly reptilian.
No, it's not an earthly reptile.
So you're telling me this is tissue from a creature from other than Earth?
From, I don't know, because this is a tissue sample from nothing that we have any data collected on Earth.
All right. You also received a blood sample, or what we are calling blood.
What we used to call, exactly, blood. Besides that skin tissue sample, we have another muscle sample, too.
A muscle sample?
It was, attached to it. The muscle, it's connected to this layer of skin, which resembles, this muscle resembles what is called the occipitofrontalis muscle. In humans, it's called tissue. It resembles, but it was kind of yellowish pink in color. In humans, it's usually red. This one didn't have that red color. In closer examination, there was another smaller, fine muscle that was intersecting this occipital muscle, resembling what is called the temporal fascia muscle. In the humans, the two are connected, and it was connected by tendinous and fatty fibrous tissue. That was similar to humans. Completely.
Well, I just want to be very clear for the audience that what we are describing here, the tissue, even the muscle sample, is close to in some respects, but different enough to be called absolutely non-human.
Oh yeah, completely.
Completely. All right.
Non-human.
All right. Now, on to the blood, or what we call the blood.
Okay, there's a sample that we call, at that time, we call it the blood. This was really interesting. The sample was dried. When you put it to the freezing, you put it into contact with really low temperatures, usually liquid tissue, because blood is considered a liquid tissue.
Right.
They get really dry, and we have to rehydrate that tissue again. We rehydrate the tissue with saline solution, and at that time we're going to be able to study the cells and if they're different. The general findings, when we tested it, reveal a level of oxygen and carbon dioxide and nitrogen-based gases, but with a slight difference. In this case, there was a high copper sulfate traces.
Copper sulfate?
Yeah, copper sulfate, really high traces of that.
Copper sulfate?
Yeah. The copper sulfate traces, there were white and red-black corpuscles what they call corpuscles with elongated nucleus.
God, I want to stop you for just a second and remind my audience that in a recent significant interview we did on animal mutilations, they found missing from the cows what? Copper. The copper content, I think in the liver, if I'm remembering correctly, was almost entirely missing. And it just keyed when you said that to me. Fascinating. So anyway, continue, please.
Okay, when we keep sampling and testing it, we look for chromosomes because we're actually working with red blood cells.
Yes.
And we're looking for more specific proofs and findings. And when we did the chromosome study, it revealed that the red blood cells, or what we could be resembling red blood cells, they have a chromosome count of 46, exactly as human.
Okay.
This is very human. We have 46 chromosomes too. But what is really, really, really astonishing is that 9 of those 46 chromosomes are not human.
9 are not human?
9 are not human, but they're only found in dolphins and some aquatic sea tortoises.
But these 9 chromosomes are found in dolphins and some aquatic sea tortoises?
Yes, yes, yes. This is the only data that we have that resemble this type of chromosome that we found in these blood.
Is there any way that you can imagine that any creature that we know of could carry such a chromosome structure?
No, that is unique. We check catalogs in many, many universities, and we went through a lot of data and trying to find some relationships between these scientific findings with any animal that we know, that science actually knows in our planet, and we couldn't find any way to fit it.